Hello and welcome to Botany 101. My name is Angela Nishimoto and I am the teacher for this class. Okay. All right, so we're going to do a little bit on biotechnology. Okay, remember the last time we talked about protein synthesis, and this is related to that, all right, and genes and all that sort of thing. So today's objectives, okay, first one is we're going to talk about DNA technology. Okay. Secondly, we're going to talk about some of the techniques that go into doing DNA technology, which is what we call recombinant DNA techniques. Thirdly, we're going to talk about DNA fingerprinting, because DNA technology does have an impact on certain kinds of investigations, okay, that we call DNA fingerprinting evidence. And then we're going to talk about genetically modified foods. They're also known as genetically modified organisms, or GMOs. Okay? So first of all, what is DNA technology? DNA technology is simply the study of and also the manipulation of genetic material. And if you recall from the previous lectures, the one before last, we talked about Mendelian genetics or classical genetics, that generally we have this genetic material, okay, in plants' genomes, also in humans' genomes. And what we do is we study this and we can actually manipulate it to our own purposes, okay? A good example of that is that we have maize. Remember what maize is? It's corn. In the United States, we call it corn. And there have been maize plants that people have genetically engineered to actually produce its own insecticide, okay? So here we have a whole field of corn. You can see the amber waves of green and the purple mountains majesty, you know? So this is probably in the US, all right? It looks like a very kind of archetypal American sort of picture here. Okay, so maize has been actually genetically engineered to produce its own insecticide. We'll talk about more of that later. And here, in this image here, this is sort of like an icon, okay, or the poster child for biotechnology. Okay, biotechnology is like the manipulation of living things, okay, for our own purposes, like we talked about. It's almost synonymous with DNA technology. Now, this is a tobacco plant, okay, a tobacco plant that has had in its genome excuse me, researchers have inserted genes from fireflies. So this plant is actually fluorescing, okay, or glowing in the dark. Oops. Another thing too is here you have these different kinds of varieties of corn, okay. And what you have is you have the production of pesticides. And here is a space-filling model of sort of like an infamous pesticide called DDT, all right? You can see that you have these chlorine atoms here, okay, an interesting thing. And here is the sort of like a skeleton model of it, yeah? So you have these chlorine atoms. Now when you think about it, so this is another image of DDT, okay? You see these ring formations. Generally the carbons will be at these intersections, okay, where they're not really notated on this kind of drawing. Right? Now, an interesting thing about DDT and many insecticides, okay, when they're very effective, they often have these chlorine atoms on it, all right? So another kind of very important insecticide that has been used for many years for termite treatments, they use it for ground treatment, or they used to, something called chloridane. Okay, so chloridane has chlorine in it too. So the thing about these different kinds of molecules, DDT or chloridane or other of these, I think they call them chlorofluorocarbons or something like that. And these particular insecticides are actually very, very effective because of their structure. And the reason why that they're dangerous is also because they're very effective. They tend to linger very long in the atmosphere, you know, whether you put them in the ground or whether you spray them over crops. And DDT, as we might know, has been warned against way back in the 1950s by Rachel Carson, who started the ecology movement, okay? Because what happens is that DDT, chloridane, lingers for a very long time. So what you can have is you can have amplification. You know, one organism taking it into their body, the next organism that eats this other organism, then taking it in, It's sort of like mercury in tuna. These different kinds of chemicals will build up in the bodies of these organisms and cause problems. And DDT, you know, one of the things that it made it like a real standard in the environmental movement had to do with the fact that it would amplify and cause problems with raptors or predatory birds, such as the bald eagle. Okay, so in the case of DDT, it actually caused bald eagles' eggs to have a very brittle shell, so that generally the baby inside of the egg wouldn't be able to survive. Anyway, so one thing, also chlordane, used as termite treatments. They don't use it anymore because it does persist in the atmosphere or in the soil. All right, and that, the funny thing was what made it effective. Some termite companies would actually warranty their ground treatments for 10 years, 5 years or 10 years, and if anybody had any more problems with ground termites, they would sometimes retreat it. Okay, retreat the ground in that particular house. So the very thing that's kind of ironic, the very thing that makes these insecticides very good, very persistent, they really work, they get high kill in terms of insects, but at the same time they're persistent, and that's also the negative side to them too. They're dangerous, okay? So here on this, on the computer, you can see a plane spraying crops. All right, so what we could do is we can actually use DNA technology to produce insecticides that are, more active. They're actually more effective in some ways, and maybe be able to engineer different kinds of insecticides that maybe make it safer for people. All right, that's the whole idea behind it. Another use of DNA technologies is that we can do DNA fingerprinting, and of course this is something that we know from the O.J. Simpson sort of trial like that. The DNA fingerprinting can actually be used to solve crimes. One of the reasons for this is that they call it DNA fingerprints because supposedly the sequences of bases making up the DNA, okay, the sequences of bases, of course, are unique to every individual. So this is one reason why they call them fingerprinting, because apparently what they'll say is that, the percentage-wise, the possibility of somebody having the same DNA sequences as another person is very, very small. They usually use it in, 1 in millions or 1 in billions. All right, so that may be true, but still, so DNA fingerprinting can be used in some cases where there is evidence such as blood or semen or any kind of body parts at the scene to help solve crimes. Excuse me. Another thing that people have used this DNA technology for is that they've actually used these DNA fingerprints in a very sad thing that had happened, was to the remains of people after national disasters such as 9/11 or even Hurricane Katrina. They were able to use, pieces of the people's, bodies to identify, victims of these various events that happened, okay? Another thing that we can use DNA technology for, we can use it to study the origins of different kinds of living things, okay? So here's an image of petrified wood, all right? People are actually able to take, DNA from some fossils, amazingly, okay? So they even have this mastodon as well. And here's another piece of petrified wood, all right? So they can actually get DNA from fossils from thousands of years ago, tens of thousands of years ago, hundreds of thousands of years ago, or millions of years ago. Isn't that amazing? So you can take it from animals, you could take it from different plant materials if some manage to survive, okay? And you can also take it from people. Do you ever remember hearing about the Iceman? He's a body, I guess, in the Tyrolean or the Swiss Alps, that they found his body, and he'd actually been in a glacier, in a frozen glacier for probably tens of thousands of years. So they're actually doing investigations as to any DNA that still survives. Because you can think of DNA as being a molecule, of course, of heredity. Remember, it's one of the kind of macromolecules. It's a nucleic acid, right? So you could actually get this fossil DNA from fossils, plant fossils, animal fossils, and also humans, as well as other animals. Okay, so you can actually get it. You can, even if it's fragmentary, even if it's only a small piece of DNA, they can still try to see if there's any difference between the living things that live now, living plants, and some of its forebears or ancestors, okay, that might have existed before. Now here we have a piece of fossil amber, and you can see there's an insect in here, yeah? So amber, of course, is fossilized tree resins. What would have happened is some kind of tree or other kind of plant would have oozed out this resin, and maybe this insect got caught in it and then died and then was fossilized in here. One of the reasons why this insect carcass can actually be fossilized is because this amber or the resin has excluded oxygen. Remember that oxygen is a molecule that helps break down other molecules. Remember that it was used in cellular respiration to break down sugar, right? Just like in the combustion engine uses oxygen to break down hydrocarbons, the gas molecules. And here, of course, is some jewelry fashioned from amber. And I think amber actually has electrical properties. I think a Greek scientist a long time ago during the ancient Greek days, they actually showed that you could rub a piece of amber against cloth and you can actually cause an electrical charge. Okay, or I'm sorry, I'm mistaking it with something else. Cause magnetism, cause things to stick to it. Okay, so generally you can get amber jewelry as well. Very beautiful. Another use of DNA technology is that it can hopefully be used to cure diseases, okay, whether in humans or in other animals or in plants, okay? So here we see a plant that has been infested with something we call powdery mildew. It's a fungal infection, okay? So what we can do is we can actually take a piece of a plant and actually insert genes into it that might make it resistant to certain diseases. And of course, there are different kinds of research going on to try to help people who have other kinds of diseases, such as genetic disorders. And one type is called SIDS, S-C-I-D, and that is when you have problems with your immune system. Do you remember the boy in the plastic bubble, this young boy that didn't have much of an immune system? I think he did later expire, he did die, but it wasn't necessarily related to his disease. I think they were able to actually successfully treat him for this SIDS, when somebody is born without a complete immune system. So generally somebody in the past who would be born with this kind of disorder, they would the doctors would pretty much expect them to expire within maybe, just within a few years after being born. But now they're actually able to take the defective gene from the bone marrow, in the person's body, and they're able to genetically engineer it or change it and then inject it back into the bone marrow, and hopefully they'd have normal cells, the normal gene in there. So what they could do is they can actually help treat human diseases as well as plant diseases and other such, okay? Now here we see a potato tuber. Excuse me. You have a potato tuber that has been infected with something called Phytophthora infestans. That is a water mold. Okay, this is actually what was responsible. The pest was actually responsible for what they call late blight. And late blight was responsible for the Great Famine or the Great Hunger that happened in Ireland in the 17 and 1800s. 1 million people died of starvation in Ireland. Another 2 million people emigrated out of Ireland and moved to Canada, the United States, and into Australia and other places in the world where they had a lot of immigrants from Ireland. The reason for that was because of this disease. And so this actually points up, for one thing, the fact that generally it can be very dangerous to have what we call monocropping. Most of the potatoes that fed the Irish population in the 1700s and the 1800s were actually of one clone. They just vegetatively propagated this plant from one place to another. So almost all the potatoes in Ireland, which sustained the Irish population. For many hundreds of years, Ireland was the poorest country in Europe, and generally potato was held to be a crop. If you eat enough potato, you can actually supply enough nutrients to sustain yourself and to sustain your family. A man could actually have maybe an acre of stony ground, even poor soils, and the Irish potato would actually continue to grow. So what you had is you had a monocrop. You had one clone throughout all of Ireland. And then when there was this pest that came and attacked the crops, okay, and what you see here is called rot. These tubers would rot in the ground, rendering them inedible. So you had the million of people who starved to death. Okay, so this points up the danger of having only one clone of a particular crop. What can happen is you can actually have a pest that comes in, attacks it, wipes it all out, and then you can have mass starvation. Okay, that is a danger that happens in this world, where we, in our modern world, rely on maybe about a dozen different kind of plants for 80 or 85% of our caloric needs. Very dangerous. Okay, so the best thing to do for nutrition as well is to eat a variety of foods. Eat a whole different variety. I know that I do this too. I tend to eat a lot of spaghetti. I tend to eat maybe bread, you know, just a few different kinds of food that I tend to like, that is relatively inexpensive and then feeds me. But the best thing to do, the best way to eat is actually eat a variety of foods. If you do it, maybe you don't even need to do, you know, vitamin supplementation, okay, which can get expensive, all right? Now, this is one way that DNA technology is used to manipulate genetic material. In some cases, we use a vector, and in this case, we're using the scientists, whoever would be doing this experiment, would be using a virus. Okay, remember that a virus is merely a nucleic acid coated with proteins, right? So if you actually can insert a gene of interest that you want to go into another kind of cell into this viral vector, the virus will actually enter the cell, the target cell, okay, would shed the vesicle that packages it, because of course part of the cell membrane would envelop the virus, and then the vesicle would actually allow the virus to be free in the cell. Then the virus will inject the DNA into the nucleus of this eukaryotic cell, and hopefully, if everything goes right, it actually seems a lot easier than it actually is. It's a very easy thing to kind of visualize, but at the same time it doesn't always work. But if everything goes right, the virus will inject it's nucleic acid with the gene of interest into the nucleus of the target cell, and then it will be incorporated into the DNA of the target cell. Then, of course, if you remember from our last lecture, then you'll actually have the DNA, including the target, the gene of interest, being used to make a protein, okay, using the new gene. Okay, isn't that remarkable? Isn't that amazing? So this is something that we humans can do. It's just like something, boy, out of science fiction, of course. Because the thing too is that when we can actually use these different kind of techniques, it depends on our imagination. Anything that you can imagine can somehow come true. That's the true thing. Another thing is that for DNA technology, they have managed to sequence a human genome. Okay, there's like 4 billion plus base pairs in the human genome. So here we have a machine, and this particular machine is one that is actually used to make DNA. It's something that you can actually construct DNA using RNA. Okay, that's usually called cDNA or complementary DNA. So this is actually showing the A, T, C, Gs that actually make up, that whole string of nucleotides that make up the double strand of DNA. Okay, we also have another kind of machine that is used in DNA technology when you want to find out whether one organism is more closely related to another organism. That's called a sequencer. Okay. So a sequencer, the person would actually, the scientist or technician or graduate student in some cases, would actually be just extracting DNA from a cell. So that isn't as easy as it sounds. You have to actually pulverize a cell to get the DNA, most of the DNA out of the nucleus. Then you might actually try to figure out what chromosomes it's on. But first, what you could do is you could put the sample in a machine, and the machine will actually give you the DNA sequence. Isn't that amazing too? Okay, so we humans are capable of all kinds of things, wonderful things as well as things that are not so great. Okay, so we use a process known as recombinant DNA technology in order to do some of the different things that we do. Okay, like we just said. Okay, going back to the slides, we have here 2 Nobel laureates. You might remember Dr. Tatum that we talked about last time. Beadle and Tatum did some work. Well, Lederberg and Tatum in 1946, they eventually got a Nobel Prize, both of them together. Okay, very famous scientists, very brilliant fellows, They did work on Escherichia coli. If you recall, E. coli is a coliform bacteria that naturally lives in our gut. Okay, it's actually beneficial. Okay, if it's in the right form. And Escherichia coli, of course, bacterium. So here, Lederberg and Tatum discovered something called transformation. Okay, so what happens is that you can actually have bacteria that are alive in solution. They will actually take up DNA that's floating around in the solution. That's called transformation. Another way that bacteria can combine genes. Nobody knew that you could have recombinant DNA in bacteria until Lederberg and Tatum did their work, and way back in 1946, over half a century ago. Okay, so they actually demonstrated bacterial transformation. Another way that bacteria can combine genes is you have something called transduction. Or let me talk about conjugation first. So you have transformation, you have another process known as conjugation. And what is conjugation? You think of, well, conjugal rights. It has to do with men and women when they're married, right? And so conjugation is when you have one bacterium and another bacterium, one designated the male, the other one designated the female. Generally, the male will form a sex pilus. It's actually a connection between the male, so-called male bacterium and the female bacterium. Bacteria don't actually have sex, okay? But what they'll do is the so-called male will send out a sex pilus and connect to the female bacterium. And then the male will actually transfer genetic material from its cell into the so-called female cell or the recipient cell. That's called conjugation, okay? The third way that bacteria can actually take up other DNA is the use of a vector. This is called transduction. So usually the vector is some kind of virus, and this is important for what we're going to talk about later. The virus is used as a vector. You'll actually have DNA, taken up from the bacterium. We're going to get into this more later, how it's actually done. It will actually, the virus will actually infect another bacterium and then inject the bacterial genome from the other bacteria into the second one, okay? That's kind of interesting. So that's called transduction. Okay, so in the 1970s, so way back in 1946, you had Lederberg and Tatum figuring out transformation, right? And then what you have in the 1970s, you have something called recombinant DNA technology. So the technology had developed by the 1970s so that we could actually combine DNA deliberately between different organisms, between one kind of organism and another kind of organism, okay? You can actually combine genes from different sources, all right? Isn't that amazing? Okay, for a lot of people, you think of Frankenfoods, you think of, mad scientists, okay? So what you could do is you could actually take DNA from one cell, okay, and put it into a different cell, okay, using vectors or using any number of different kind of techniques. So here we have a bacterial cell, and here we have a depiction of the bacterial chromosome. Remember that bacterial chromosomes are circular, whereas eukaryotic cells, such as plants, such as animals, such as fungi, such as protists, we have linear chromosomes, okay? So bacterial chromosomes are circular, all right? And here you can see that bacteria can also have little pieces of DNA that are circularized also, and these are called plasmids. Okay, plasmids. Now when a bacterium, so usually bacteria, what they do is when they grow, what we could talk about when we talk about bacterial growth, we're actually talking about bacterial replication. So one cell, okay, becomes two cells, okay, and what happens is that when the bacterial cell replicates, it'll actually also replicate their DNA and also in their circular chromosomes, and they'll also replicate the plasmids in many cases, okay. So you have the bacterial chromosome, the circular chromosome that you see here in green, and you have the plasmids seen here in red, okay? Another thing can happen is that you can have a bacterial chromosome, okay, with a plasmid in the same cell, and then the plasmid can be incorporated into the bacterial genome in this way, okay? That's what they call an episome, when you actually have a bacterial chromosome that has plasmid genetic material in it. Okay. Then what happens is that cell replicates, it will actually form two cells that has the episomes in it. Isn't that remarkable? Okay, so this is part of the basis of what we call DNA technology. You can actually incorporate DNA from another source, like this plasmid might have come from a different source, into the bacterial chromosome. Okay? Okay, so here we have another depiction of the circular chromosome, and in this one it's all shown to be all bent and folded of the bacterial chromosome. And then you can see you can have lots of plasmids in a bacterial cell. Okay, this is actually a little bit more a realistic depiction. Okay. Okay, so in the 1970s, okay, you had this recombinant DNA technology, and what you could do is you could actually combine genes, okay, from one source and insert the DNA into a different source, so you have genetically modified organisms. They've actually been changed by us humans deliberately, okay, such as this very famous image of the tobacco plant fluorescing with the genes, you know, the expression of the genes of a firefly. So a firefly, of course, is an animal. Animal genes put into a plant, and here you can have its expression. Okay. Another thing that some of you might remember too is at the Bishop Museum, they had a display of these mice that glowed in the dark. Apparently what the people did, the scientists up at UH, what they had done, they'd inserted genes from a jellyfish, a fluorescent jellyfish, into these mice. Okay, so you had glow-in-the-dark mice. Okay. Anyway, so what you have, so we do have the copying of genes, okay, in the new cell. And another thing that can happen is that these genes can be expressed, because you recall the last time when we talked about molecular genetics, okay, remember that the gene, okay, is the whole basis of protein formation. Okay, remember in terms of DNA, so you have a gene on DNA, DNA codes for RNA, right, messenger RNA that takes a message that is made to use to make a polypeptide. Okay, so remember the whole thing, you have transcription, right, where DNA codes for RNA. Then you have translation, where the RNA will code for a series of amino acids making up the polypeptide. Okay, so this is like magic. Okay. Now, one of the first type of, so what we're going to cover next, we're going to go from humalin, which is human insulin produced artificially, to later we're going to talk about genetically modified foods. Okay. All right, so in 1982, so generally from the 1970s into the 1980s, DNA technology advanced and advanced and advanced. So then we had this huge company called Genentech. It was the first company in the United States to actually patent a way to actually produce humalin. Okay, humalin is synthetically produced human insulin. So how did that happen? Okay, so what they did is they took human genes and inserted it into bacterial genes, all right, into the bacterial DNA. Okay, so these genetically modified bacteria were grown, okay, in these huge vats, and they produced this humalin. So before these genetically modified bacteria were actually produced before 1982. Okay, people had to get their insulin. People who had diabetes, type 1 diabetes, who need to inject their insulin, they had to get their insulin from, I guess, from pigs and from cattle, bovine insulin. And of course, when that happens, you know, humans can often get a really bad reaction from injecting, you know, foreign substances into their body, even something that their body needs. Okay, so that was very dangerous for people who had problems with their blood sugar, you know, who had diabetes. Okay, so they were actually able to genetically engineer these bacteria to produce a human protein, okay, human protein called humalin. Okay, so this is a couple of different kinds of illustrations of insulin. Okay, you can see that it's a kind of well, it is pretty large of a molecule, but still, think about it. So even think about the miracle of our genome being able to produce something as complex as this. The miracle is that in most people it works, okay? Our bodies work for the most part, okay? And yet people who can't produce enough insulin or can't produce it in a way that can help regulate their blood sugar, we actually have technologies now where they can actually inject human insulin produced by bacteria. You have to imagine what an amazing thing this is, okay? Now another kind of hormone, or another kind of human product, is HGH, human growth hormone, okay? People have been able to engineer, okay, so this is a model of growth hormone. So in the past, you know, you couldn't get growth hormone from pigs or cattle or sheep or any other kind of animal. People actually who had problems with, like if they were very tiny and their bodies weren't producing enough human growth hormone, what they had to get their human growth hormone from, they had to get the human growth hormone from cadavers, from people who were dead, okay? So as you can imagine, these injections of human growth hormone were extremely expensive, very hard to get, Okay, and also, you know, if you actually got it from the wrong person, you know, you could actually probably get diseases from them too, okay, depending on what they died from it and etc. Anyway, so now DNA technology has produced these miracles where people can actually get access to human growth hormone that is grown by a different organism, not humans, all right? I believe that these are actually produced by fungi, okay? Another kind of product of DNA technology and recombinant DNA technology, we're actually able to get vaccines, vaccinations. What are vaccinations? Generally, vaccinations are, say, different kinds of disease-causing organisms, such as viruses or bacteria. Okay, so what they'll do sometimes is they'll kill or heat kill, you know, the bacteria or render the virus ineffective, and they'll inject you with this dead bacteria or pieces of the bacterial proteins, or they'll give you pieces of the bacteria itself. And this will provoke an immune response in our bodies, okay? This provokes an immune response and a production of antibodies, all right? These antibodies will then, if you're infected again, okay, with the same agent or a similar agent, then your body will rally its defenses really quickly. So it's not so likely that you'll get a really bad infection from it. It'll render you immune. Okay, it'll actually adapt your immune system and render you immune to these different diseases. A lot of people nowadays, maybe too, you know, they might, you know, have a thing about it, and it might be true in some cases, okay, that vaccinations might actually cause some diseases. That's true. When I took a flu shot, okay, when I was a student, and that particular day when I took the flu shot, I'd actually not had very much sleep. Yeah, I was running all day long, lots of stress, you know, because I had classes, plus I was teaching at the same time. So it was very difficult. So from that the flu vaccine that I got in my arm, of course, I actually got really sick from that. Okay, so that can happen. That's definitely a risk that you take whenever you get a vaccination. Some people even allege that maybe the very large, kind of larger numbers of children now with autism or other sorts of, you know, disorders, some people think that maybe some, you know, vaccines might have caused it. I don't know if that's true or not, but it's also, you know, some things that you have to take into account. Another thing to remember is that the kind of vaccines that we have are generally against or for diseases that are very, very dangerous, that if people get these diseases, they're very likely to die. You know, diphtheria, whooping cough, smallpox, which people don't take anymore because apparently smallpox is supposed to be eliminated. Polio is another disease, very bad diseases with very bad effects. It can kill children and other people. So you yourself, when you become a parent, or if you are a parent, you have to kind of weigh in about what do you think are the risks. Most people would opt to get their children vaccinated just because the diseases are so bad, okay? Generally, you don't want your children getting that. Okay, so most people will opt to get the vaccines. Besides which, I think for a lot of schools, they won't let you enroll your child in school unless they've been properly vaccinated with certain vaccines. Okay, now we also have the idea of genetically modified foods. All right, okay, so in the year 2002, okay, approximately half of American corn or American maize and soybeans were genetically modified, okay? So there's a whole bunch of controversy in that too. And so this is one, you know, example. This is actually a leaf of corn, and it has a corn borer on it. Remember earlier we talked about that we actually had corn or maize that had been genetically modified to produce its own insecticide. That's true, okay? They actually had corn that was genetically engineered to produce its own BT. It's actually Bacillus, I believe, Bacillus thuringiensis, okay? What it does is actually something that will cause an infection in some insects, okay? What is it? It's a bacterium, right? So what it would do, it would actually attack the insect and pretty much make holes in it or kill it in some way, all right? So what happened is that with these different kinds of corn, the controversy about it, so it was good because it would kill this corn borer and actually produce fruits and ears that would be very healthy looking. And this corn borer actually makes for a really bad, you know, really eaten sort of corn ears. And so what happened is that they found out that some of the pollen from the corn was actually getting onto other kinds of plants, okay? And of course, the corn pollen couldn't actually fertilize these other plants, okay? Milkweeds, I think, in particular. Now, can you think of a particular kind of insect that feeds on milkweeds? Okay, so think about that. Okay, you have these butterflies, right, that will feed on milkweeds, the monarch butterfly caterpillars. What they'll do is they'll eat the milkweed, remember, because milkweed has this kind of sap in it that is, supposed to be poisonous to some insects, but the monarch butterfly caterpillars can eat it. So what they're finding out was that the BT pollen, the BT, okay, that was in the corn, the pollen was actually getting onto the milkweed and rendering the milkweed poisonous, or the pollen that was on the milkweed was being eaten, okay, by these larvae or caterpillars of the monarch butterfly. Okay, so it was killing them. So that was something that was pretty controversial and pretty alarming at the time. So, so the thing too is that even though about half of American corn and also soybeans, okay, so here we have a picture of soybeans. Soybeans, also known as edamame here in Hawaii. And so the soybeans usually is used, and a lot of corn that is produced too, is usually a lot of corn and a lot of soybeans are actually eaten by cattle. Okay, and pigs and other kinds of domestic animals that we use for food. Now soybeans are good too. Actually, soybeans are supposed to be very good to eat, okay? Because I think in Asian cultures, okay, what the edamame I think is a Japanese word for soybeans. So I think they have them in bars and things like that too. If you go out, sometimes they'll have soybeans in the fruit, and they'll boil them, put a little salt on them. Very good eating. A thing too is that soybeans actually are a source of what they call phytoestrogens. Okay, when women get into their 30s and into their 40s, hormone production starts to drop on different types. Okay, and what people can do is they can actually eat soy. So a lot of times when you go to the grocery stores, you'll see a lot of maybe older people or older women or people, people who are in, women who are in their 30s and 40s, they eat tofu or they eat other products of soybeans, because of course soybeans actually are used to make tofu. And that actually has what they call phytoestrogens. Phyto just refers to plant and estrogen refers to the hormones. Okay, so when women are getting up there in age, it's very good for them to eat soybeans, among other things, like yams. Okay. Another kind of genetically modified food is called golden rice. All right. And here we have white rice, yes, and here we have golden rice. They've actually inserted genes in the rice plant to produce rice that has beta carotene. Now, really altruistic sort of motives kind of powered the move to produce this golden rice, because in some places where people rely very heavily on rice, even if it's brown rice, this is of course hulled rice, right, white rice, and so that when they eat a lot of rice and don't eat a whole lot of fruits and vegetables and other sources, they can actually get a deficiency in vitamin A. Beta carotene is a precursor of vitamin A. All right, you can actually eat quite a bit of beta carotene, okay, and actually excrete some of it too. But they say don't take a whole lot of vitamin A, because vitamin A is oil-based and it can build up in your tissues, and you can have bad effects from that. Okay, but if you actually, so some people who don't have enough beta carotene in their diet, you can actually go blind. You can actually lose your vision. And in some places, especially in the third world, where they have a lot of people who are poor, okay, you can actually have blindness as a result, because if somebody's poor, if they can't afford to buy food, nutritious food for their family, well, very often they can't afford medical treatment either. Okay, so beta carotene in rice, golden rice, is something that people are producing, and also people are growing in certain places of the world. I think in some places in Asia, yeah, and other places that that they rely a lot on rice. Okay. So that's like a success story, I guess. But one other thing too is that there's also controversy about this as well. Okay. First of all, we're going to talk about recombinant DNA techniques before we get into some areas of controversy. Okay. Remember that you have bacterial plasmids, small pieces of DNA that are circularized. Okay. So sometimes they use bacterial plasmids to actually move genes from one organism into another. Okay. And they're used, and when they're used in this fashion, they're called vectors. Okay, just like you can say a vector for disease, like malaria, would be the mosquito, right? It's an animal or a living thing or something that actually moves, the mycoplasm from one person to the next to transmit and that mycoplasm is what causes, and its life cycle is what causes malaria. So you can actually use these bacterial plasmids to move genetic material from one bacterium into another. Okay, remember transduction that we just talked about a few minutes ago? So in these bacteria, you can have the foreign gene replicated in these from these vectors. You can have the foreign gene copied by the bacteria, right? Bacteria. Okay. Another thing that you can do is that this is deliberate, all right? You can have a particular desirable gene in one organism. You'll use something called restriction enzymes. So what are restriction enzymes? Bacteria, okay, in their bacterial body, you know, the single cell bacteria, which is what they are, they'll actually have enzymes in there called restriction enzymes. What do you call them? Restriction enzymes. Restriction enzymes, actually what they'll do is they'll restrict foreign DNA. Okay, what they do is that they'll cut up this foreign DNA. So just like in our bodies, you know, generally if we have something invading our bodies, our immune system all kicks up, and what they'll do is they'll produce antibodies to repel these invaders, right? So what these bacterial cells do, they actually have these enzymes called restriction enzymes. What they'll do, they'll cut up foreign DNA, okay? So we humans, we figure out a way that we can use this in our technology. Okay, so here, over here on the slides, you can actually see a little model of an enzyme. Okay, so what restriction enzymes do, they'll restrict foreign DNA, they'll cut them up. Okay, and here's another model of another enzyme, right? Okay, just to show you enzymes, remember enzymes are proteins, and proteins are very large, very complex macromolecules, right? So this is how enzymes work. Okay, so here you can see that the enzyme itself is pretty big. You know, this one is not as big as some of them, but here you have an enzyme, right? An enzyme helps to facilitate chemical reactions. Now here you have the molecule that the enzyme is going to have its effect on. That's called the substrate. Okay, so generally remember that you have the 4 levels of protein structure, right? You have the series of amino acids, you have the bending and folding and hydrogen bonding of nearby amino acids, and the 3rd level you have the bending and folding and 3-dimensional conformation of the molecule depending on the hydrogen bonding and in far apart amino acids. Okay, so the 3-dimensional structure of an enzyme actually determines its function because it has these binding sites, these active sites on the particular enzyme. And what you have, the substrate and the enzyme fitting together. Okay, you have something called induced fit. Okay, so what happens is that enzyme will actually change the shape a little bit so that the substrate can fit into it snugly. Okay, the next thing that happens is that the chemical reaction is facilitated by this enzyme, and then you have the substrate, okay, or the reactant actually turning into the products. Okay, so in this case, the bond is being broken. Remember that that is called catabolism, when you have a larger molecule becoming smaller molecules. Okay, then the enzyme releases the products and then is ready for the next one, because an enzyme generally is not changed after it actually causes a chemical reaction in its substrate. The enzyme remains unchanged. Okay, so that pretty much is how an enzyme works. If you're to try to figure out anabolism, where you have smaller molecules becoming a larger molecule, okay, it'll be sort of like the inverse of that. You'll have the reactants, you know, binding onto the different sites. You have a bond forming between the reactants and eventually becoming a product, which is larger. Okay, in anabolism, when you have smaller molecules being put together to build a larger molecule. Okay, so this is how enzymes work. It looks very mechanistic, you know, and that actually is something that happens, okay, in our bodies, in the plant bodies, all the time. So when you get into biology, you know, when you really see the beauty of it, it's like a miracle, every single bit of it, you know, okay? All right, then, after the restriction enzymes in recombinant DNA technology, after it cuts the DNA that you want it to cut, okay, you have another enzyme called DNA ligase. This is sort of like a little bit gory, but you can remember DNA ligase from when you think of those CSI, all those different television shows, okay, you think of ligature marks. What are ligature marks? It's when somebody's been throttled, right, with a rope or something, right? Ligature marks. So you think of ligase to have to do with tying together, okay? So DNA ligase will then paste, okay, this DNA into where you want it. Okay, so we're going to see this in just a sec. Okay, here. So when you cut the DNA, you're going to actually cut DNA in one organism, or you will have extracted the DNA first, okay, from one organism, get the DNA from another organism. And you use the same restriction enzyme to cut the DNA. What happens when the DNA is cut? It usually cuts it at a particular site, okay? We'll actually cut it at a particular site, okay? So one of the things is that you'll have one overlapping part of one strand of the DNA. That's called a sticky end. This is really true. I'm not joking. It's called a sticky end. Okay, so you're going to use the same restriction enzyme on both DNAs when you're doing this recombinant DNA technology where you're combining the genome or part of a genome of one organism with another one. Okay, so before DNA ligase, you'll have these two, pieces of molecule with these sticky ends. And one of the things you'll notice is that they are complementary to each other, right? CTACG. GATGC. They're complementary to each other. So when you add DNA ligase to the solution, you'll have pasting. You'll actually have these two parts of the two DNAs joined together. So you have two different DNAs from different sources, but you will have pasted, one foreign DNA piece onto another. Isn't that amazing, right? Okay. Now the next thing we're going to talk about when we talk about DNA technology and also biotechnology, okay, it's called DNA fingerprinting, all right? Okay, so DNA fingerprinting then is used in some cases like in criminal investigations, also things like finding out if somebody, is a father of a baby that he claims isn't his baby, So if it is actually his child, they can actually do something called DNA fingerprinting. Okay, so how does that work? Okay, for one thing, all the DNA of every single person, every single plant, every single animal is unique, okay? Unless you have an identical twin, okay, your DNA sequence is unique. There has never been a person like you ever in the history of the earth before you. There will never be a person exactly like you in the future, okay? Your DNA sequence is entirely unique. And because it's unique, okay, then you're going to actually have different DNA fragments depending on the person. Remember that your DNA actually codes for what you look like, what kind of chemicals your body produces, what kind of biochemistry goes on, okay, in your body. Okay, it has everything to do with your appearance, all the different traits that make up you as a person. You know that you are unique. There's nobody exactly like you in the whole world, ever before, ever after. Okay, you're completely unique. And if this is true, okay, if this is true, what they'll do is we'll actually take, okay, your DNA, and you can actually cut it up with restriction enzymes, okay, and then amplify it in some cases. Okay, so now before we get on with that, we'll talk about something called the polymerase chain reaction. This is a process invented by somebody named Kary Mullis, okay, in the 1970s. I believe he was working out of a university somewhere. Anyway, so he invented this technique called the polymerase chain reaction, also known as PCR. What happens during PCR? Okay, remember that a DNA molecule is two strands, right? Two strands hydrogen bonded. Remember when you have this hydrogen bonding, which is a weak kind of bond, right? It is a weak kind of bond and it can be disrupted, okay, by heat. It can also be disrupted by changes in pH. So what they'll do in the polymerase chain reaction, think back on when your DNA is actually being replicated or the plant DNA is being replicated, what happens? The DNA unwinds, right? The DNA unwinds, and then you have these free bases coming in and joining, to these free strands, and then you will get two strands of DNA from one strand, right? Remember when we were talking about DNA synthesis that happens during the S phase, before we have mitosis or meiosis, right? Do you remember that? Okay, so the polymerase chain reactions takes the actual DNA molecule and it actually uses the properties of this molecule to allow these chemical reactions to happen. So what happens during the polymerase chain reaction? You actually use a machine called a thermocycler, okay, which is a pretty small machine. You can have one over on your desktop, maybe a couple of feet by a couple of feet big. Okay, what happens is that in here you'll have these little tubes of the DNA you have extracted. Okay, and in this you have like a little cocktail. You have nucleic acid, different kinds of nucleotides, DATPs, DTTPs, DCTPs, and DGTPs. So you have these nucleotides in there. You also have a buffer usually that helps to resist changes in pH. And you also have, let's see, what else? You have something called a primer, an RNA primer, because generally you can't make new DNA, or a new DNA strand without something to make it go. Okay, so you have these RNA primers. So you have this in there, okay, and then you also have your DNA sample. And so in these little test tubes, you put it in the thermocycler, and then what does a thermocycler do? What is thermo? It has to do with heat, right? Cycle, it runs on a cycle. So the thermocycler will also have an enzyme, okay, usually some kind of a DNA polymerase. And in the day when I was going to school, they called it TAC, Thermophilus aquaticus, okay, Thermophilus aquaticus. That's called TAC. It is actually a DNA polymerase that can resist and actually work at very high temperatures. So what do? They'll actually open up the strands of DNA by the machine actually getting really hot. So the strands will open up because the hydrogen bonds are weak. So if you actually apply heat to it, they'll open up, okay? When that happens, when they open up, okay, then you cool it down really fast. As you're cooling it down, the primers will anneal to these single DNA strands, and then you'll have the bases coming in, building up a new base. Okay, so with the polymerase chain reaction, you can actually theoretically start off with one piece of DNA. Okay, and then after one cycle for heating and then cooling, you'll actually end up with 2 strands of DNA. The second cycle after that, you heat it and you cool it, and then you'll have 4 strands of DNA. Okay? Therefore, you can have up to billions of copies for one strand of DNA made in hours. You cycle up, heat, then you cool down. You cycle up, heat, you cool down. Okay? So that actually leads us to the thought of exponential growth. Remember, you start off with 1, you end up with 2 after the first cycle. You start off with 2, you end up with 4. Okay, so here, this is actually a good example of what we call exponential growth. You know that old story where they say, would you rather somebody give you $1 a day for 30 days, okay, or over 30 weeks, okay, or would you rather have somebody give you 1 cent in the first week, okay, and give you double that in the second, double that in the third week, etc.? Okay, so if you actually take the first option, well, somebody gives you $1 a day. Okay, starting at day 0, you have $1. Okay, and on day 1, you'll have 2, day 2, you'll have 3, and etc. Until on the 15th day, okay, on the 15th day, you'll have $16. Some people would have consider that, oh, you start off with $1. That's a lot more than 1 cent. That's 100 times more than 1 cent, so I'll take that option. But if you take the second option, this is amazing. Okay, 1 cent becomes 2 cents, before it becomes 4 cents, becomes 8 cents, and then you'll have, what is it, 16, 32, blah, blah, blah. By the time you get to the day 13, you'll have about $80. By the time you get to day 15, you'll have over $300. So this is actually what they talk about when they're talking about exponential growth. Okay, when you double every time you actually go from one unit to the next. So by the 30-day point or the 30-week point, okay, what do you have? You'll probably have thousands of dollars. Okay, so this is also the miracle of compounding. You know, when you're in your 20s, when you first start earning money, if you make it a habit to actually put away a little bit of your money, okay, every time you get a paycheck, you know, you're thinking, oh, you know, it's only 10%, you know, I can start saving later. But if you actually allow the miracle of compounding, as a particular, you know, financial guru talks about, the miracle of compounding, if you start putting away money when you're in your teens or when you're in your 20s, when you first start working, by the time you're ready to retire into your 40s and into your 50s, you'll have a huge nest egg, you know, because what happens is that the interest that actually goes, that the bank or whoever you invest in will pay you, okay, will compound, and your principal grows and grows. I don't know if they call it a principal, but it will grow and grow and grow. So by the time you're in your 40s or in your 50s, you'll have like a nest egg of, you know, maybe several $100,000. That's amazing. Okay, that's something that you want to make sure that you want to do. It doesn't actually cost much. And the thing is that if you get like an automatic payment deduction, you know, before they pay you, they just take it out, you won't even miss it, you know. And a little bit at a time. It's sort of like a drop wearing away at rock. They call water the strongest element. You can just have a drip, drip, drip. But if you have enough time, it can actually wear away a huge boulder. Okay, that's why they say in certain philosophical things that water is actually the strongest element. Okay, so think about that. Okay, think about that and maybe try to apply that. This is true. You don't want to be in your 40s and 50s and have no money saved. You know, that's really true. Okay. Okay, so the DNA sequences of different organisms are unique, okay? Because of that, remember, so we're going back to the thing about DNA fingerprinting, okay? So what they can do, they can actually get a little bit of DNA from a degraded DNA from blood or any kind of body things at a particular area, okay? Even when we were talking about a little earlier, 9/11, they actually got DNA from people's hairbrushes, from their toothbrushes, because when you're brushing your teeth and your gums, a little bit of your cell goes on your toothbrush, things like that. So even if you actually lick an envelope and then you send it through the mail, if they want to find out who sent something, they can actually get saliva and pieces of your cells on the envelope, and they can actually do this. They can actually take the DNA sample, even if it's degraded. They can amplify the DNA. That's what they call amplifying, when they actually make one DNA piece into more and more and more. They call it DNA amplification. Okay, so they can actually do that. They can amplify the DNA, and they can get a big enough sample so they can actually do something called restriction fragment length polymorphism. Okay, they call them RFLPs. Okay, because what happens is that your DNA is different even from your brother's DNA. Okay. So what they'll do, they'll actually take DNA, from a suspect or something, or from a crime scene, and then they'll cut it up with a particular restriction enzyme, either 1 or maybe 2 or 3, and that'll cut at a certain sequence. And in our DNA, our human DNA, there are lots of what they used to call junk DNA, or a DNA that wasn't doing any coding. But because you have different numbers of these repeating parts of DNA, they can actually tell, that one person will have maybe 2 cuts in one small piece of DNA. Another person might have 3 cuts in one small piece of DNA. Okay, so you have these, the one with 3 cuts in it will give you 4 pieces, so some of them will be different sizes, right? So you have 4 pieces of DNA, maybe smaller than the one with 3 pieces of DNA, right? So then what they'll do is they'll get this DNA, they'll run it on a gel, okay, on an agarose gel, okay, and they'll do something called electrophoresis, okay? So when they do this electrophoresis, you'll actually have some kind of like a phage of some kind that actually will give you these banded segments. And when you look at this, you can actually see these are just having to do with the size. Okay, so there's restriction, fragment, length, polymorphisms. Polymorphisms mean that you have different forms. Restriction, fragment, so this is using restriction enzymes to fragment the pieces of DNA, okay, to different lengths. So you see that in different people, you'll have different banding patterns just because certain pieces of DNA will have been cut differently. You'll have different size pieces. The smaller pieces migrating through the gel faster. Okay, so you can see that these different kinds of DNAs actually have certain bands in common, but you can tell that these are from either different people or different samples from different kinds of organisms, because the size of the fragments are different sizes. So you can see that even here, in this column here, if you look very closely, you can see that these are like thousands of base pairs, 2,072 base pairs, 1,500, so you can see that the smaller the fragment, the faster it migrates to the gel. That's why they call them restriction fragment length polymorphism. So they can actually use this in terms of criminal evidence. Now, this is a process that was, I guess, invented by a professor. I believe he's in England. His name is Alex Jeffreys. Okay, he actually invented this technique. Okay, this is miraculous. Isn't that neat? Okay, so we can actually do this kind of puzzling and figuring it out. Okay, all right. Now, another thing that is kind of like a controversial sort of thing too, is that generally, okay, because we can actually do this, okay, because we can actually do this restriction fragment length polymorphisms, should we actually make a data bank of everybody's DNA? Okay, is that true? Should we actually do that? So there are, arguments for both sides, of course. Say for one thing, if you do have the data bank of everyone's DNA sequences or everybody's, RFLPs, right? You think, well, that could really help you track and keep track of criminals, for one thing. Okay, that's actually something we could do. We could keep track of people who do, certain bad things and maybe have been shown to have done it more than once and done nasty things to other people. You can actually find and keep a data bank of that, and you can actually trace where these people who do criminal acts, where they are. Because even now they have like a place, like a bank. They actually have a list of sex offenders, like that. They'll actually have sex offenders, actually some of them have to report where they are all the time, okay? that actually is something, I guess because one of the things that you have to weigh when you're thinking of these things, the public good and the public safety against the rights of a particular individual. So in some cases, they would consider that the good of the community or the safety of the community outweighs the rights of a single individual person's rights. They can think of it sometime. But at the same time, like you think to yourself, do I really want my DNA sequences to be known by any kind of agency? Such as, like, wouldn't there be maybe, because even now, in some people's health insurance, they'll actually say, okay, they will actually eliminate, like if you have one insurance plan, you transfer another insurance plan. Sometimes what they'll do is they'll actually say that they will not cover you for what they call pre-existing conditions, okay? So if you have a genetic disease, okay, that has been diagnosed, okay, wouldn't they maybe discriminate against certain people who have certain genetic diseases that are actually very expensive to treat, or that calls for very expensive drugs? You know, because some new drugs, it costs like thousands of dollars a year, tens of thousands of dollars a year to treat certain conditions. So wouldn't that be kind of opening up, you know, a whole can of worms, so to speak? Okay. So one of the things too is that can they actually do that? Keep track of your genes. Wouldn't that maybe call into effect someday in the future if maybe you have a predisposition for a certain kind of disease? Would certain jobs, you know, would you that close you off for certain jobs? Isn't it also an invasion of privacy as well? Okay, so you have to kind of, there always seems to be a balance when it comes to be laws or different kinds of different sorts of, you know, keeping track. You have control and safety, and you have freedom, okay, or maybe not so safe. It always comes down to that, you know, in terms of an ethical question. And also in the United States, we're supposed to have the right to privacy. You know, how would you like your DNA sequence to be put up on the internet for people to look it up and all this kind of stuff? This is not something that is totally unimaginable. Okay, it can happen. All right, so you have to kind of think about this, because this is something that we are all going to be voting on in the future. Okay, I'm thinking too, this might not be true. It's just conjecture on my part or on other people's part. We think that if somebody goes into the military, what do? You get a physical, you get all these health things. They take your blood, okay, to find out your blood type. And if they're doing that, you know, maybe they're also even now or maybe even in the future, they might actually try to be sequencing people's DNA, because your white blood cells, okay, has your genome in it. It actually has a nucleus, right? Your white blood cells, your leukocytes, right? They have your DNA in there. So what could be that they're doing that now? Because they're saying that they're trying to maybe sequence people's genome, or at least finding particular markers for people, just in case somebody dies, you know, in service to their country, and then you only have maybe parts of somebody's bodies left, you know, with all this kind of terrible, you know, techniques that we have for killing people, you know. Okay, so things to think about. Okay, I know I probably just scratched the surface. I'm sure that I haven't really gone into all the different kind of ethics and the different sorts of things that we can think about, okay, about this, right? But still, interesting things to think about. And we're going to have to be making decisions about this in the future. Okay? Okay, now another thing too. Okay, we have another issue called genetically modified organisms or genetically modified foods. Some people call them frankenfoods. All right? Now in, let's see, in 1999, all right, you know, we had a big controversy. The UK and the EU, the United Kingdom and the European Union kind of showed that they had concerns, okay, about the possibilities of genetically modified organisms, okay, whether we can actually eat these things. So here's a flag, you know, this is called the Union Jack, and we can recognize this too because this is part of our flag, our Hawaiian flag. Okay, this actually makes up a corner of our Hawaiian flag. This is the flag of the European Union. So they express many concerns about the United States, you know, making these frankenfoods or these genetically modified foods and shipping them into Europe. And I believe at one point they actually had an embargo. Okay, they actually had a limitation. They were actually trying to get the United States to maybe put labels on genetically modified foods. And of course, a lot of farmers, a lot of people that are involved in these sorts of things, they were like, very much thinking, oh, you know, if people knew that we were labeling if we labeled these genetically modified foods, people wouldn't buy them, you know. So it makes you kind of think, if people won't buy them, are they good, you know? Because the thing is that these different kinds of genetically modified foods have not really been tested over 2 or 3 generations. But of course, you know, we're kind of serving as a guinea pig. I myself, as a botanist and interested in molecular biology, I don't know I don't know whether genetically modified foods are bad for you. I don't know that that's a fact. I don't know if they're good for you either, though, you know? So this is another thing that we have to think about. Are we going to do that? Okay. Now, also, in the United States, who actually oversees these things? There are 4 different agencies that actually oversees and has oversight over the genetically modified foods, including the Food and Drug Administration, the FDA, the Environmental Protective Agency, or the EPA, the National Institutes of Health, or the NIH, and also the DOA. And that doesn't mean dead on arrival. That means the Department of Agriculture, okay? So here we have the symbol for the FDA, the symbol for the EPA, okay? So these are the governmental agencies that has oversight over these issues. The National Institutes of Health, okay? Guess they're headquartered in Atlanta, Georgia, okay? Anyway, in terms of what we're going to finish up on, so we've kind of finished today's lecture, give us a lot of things to think about, okay? So first of all, DNA technology is here. It's been here since the beginning, from the 1970s, okay? Built on a lot of the foundations, a lot of which we talked about, including classical genetics, molecular genetics, a lot of the early workers in the 20th century, and this is now. This is happening here. It is happening now in Hawaii, even as we speak. We have the College of Tropical Agriculture and Human Resources. We have agricultural engineering. We have molecular biology in Hawaii, okay? And Hawaii is actually one place, you might have read the newspapers, where a lot of the DNA technology is being used here to engineer new kind of food crops, okay? Another thing is recombinant DNA techniques. I know some people really don't like that idea. You know, could it be like we humans are trying to, you know, make things in different images? Because recombinant DNA techniques are also being used to engineer new organisms that never existed before we did. So is it we humans, you know, playing God? Is that true? Is it not? Okay. Okay. DNA fingerprinting. Should we actually make a data bank of people's DNAs? Okay, so that could be a good thing, but it could also be not so good, depending on what. So Big Brother is here, is now, all right? Lastly, genetically modified foods. Are they good or are they bad? I myself, I don't know. I might have an opinion, but that doesn't mean that I'm right. Okay, so this is what a college education is supposed to teach you. It's supposed to have you be able to have the ability, okay, to actually take in data from different sources and make up your own mind. This is one of the great things about living in a free country. Everybody has the right to have their own opinion. Okay, everybody can get their data. So as you get educated, as you continue your education, as you're learning more and more about things, more and more your judgment will be honed, and you can make up your own mind about these sorts of things. Okay, but isn't it a great thing that we have choice? Okay, that's one thing that we have in a free country. We have choice. Maybe too much sometimes. Anyway, so that's all that we have for today. Okay, it's quite a lot of things to think about, a lot of things to kind of mull over and really kind of think about and come to your own conclusion. Anyway, so we'll see you next time, and I guess we'll say aloha now. Bye-bye.